Okay, we are now going to continue with what we were talking about last time, namely using everything we've learned in the class so far to understand molecular machines. We looked a little bit at molecular machines last time. We compared them to macroscopic machines and looked a little bit at this particular one, kinesin, whose job it is to actually pull cargo down microtubules and therefore to generate forces in its own right. It's a hand-over-hand walker, remember, and it uses ATP as its fuel. We'll talk more about the nature of the chemical fuels that these machines use just now in a minute. And we looked a little bit at how it moves actually in a video, and then how that all works with this sort of mechanism. This is really a form of a kinetic mechanism. We'll look much more closely at what that means in a minute. But now what we're going to do, that was all sort of general and vague. Now we're going to look at one particular machine, the DNA polymerase, which is one of the molecules that was in the movie we showed earlier. Right, in that movie, this is the whole replosome. And it has two DNA polymerases, one there and one there, one that synthesizes the forward-going strand and one that synthesizes the reverse strand going backwards. But we just want to look at one of those machines. The replosome itself is too complicated. We're going to ask the following questions. First of all, how does the machine work? And that means understanding its structure, because you can't understand anything about a machine until you know how it's built, what its parts are, how it moves, and that's some sort of machine cycle, and that's going to be a kinetic mechanism. So right away, kinetics is really important for understanding how all these things work, how all these little machines and doodads and motors and twirlers and cutters and whirlers and pullers work. And then we'll look a lot, of course, at the energetics, because that's the thermodynamics. That's also the driving forces. That's what the fuel is used for. What causes the parts to move, what generates overall movement of the polymerase on the DNA. The polymerase generates forces. It has to. It's got to reel the DNA in as it's going along, or else the other way to put it is that it's got to move along the DNA like a little car, in a sense, a little slider along the DNA. And it has to generate at least some force for that. In fact, it generates a lot of force, it turns out. And we want to know how that works, or get some idea how that works. So that's one general patch of questions. The other patch of questions is, how does the DNA turn out to be so dang accurate? It's called fidelity is the right term. So the DNA is copying. So there's one strand of the DNA, that's the template strand. And then there's the other strand of the DNA, that's the one that's being created by the polymerase. That's called the primer strand usually. The polymerase sits more or less here. Polymerase moves along, but it's got to copy the bases on the template strand, right? And it's got to do it accurately. Every time it makes a mistake, every time it puts a wrong base in here, it's a mutation. And the next generation has that mutation forever. And if you have too many of those, you get what's called an error catastrophe. It means that as generations go by, one after another, mutations build up, and pretty soon your genome's just worthless. All the information has degraded away, and it's no longer a genetic code for a particular functional organism anymore. Also, it turns out that fidelity can be really high, 1 part in 100 million mistakes. And that's not surprising from a functional point of view, right? If you want to avoid error catastrophe, and if your genome has 100 million residues in it, most don't. I mean, even bacterial genomes, which are pretty small, have 3 or 4 million base pairs. But even so, the fact that it can reach 100 million, E. coli's polymerases can reach fidelity of 10 to the 8th, that means E. coli doesn't want any mistakes when it's copying itself. It only wants one mistake in roughly 100 generations. And so it's put a lot of effort, so to speak, into making its polymerases really copy correctly. Well, how do they do that? 1 part in 10 to the 8th is a phenomenal level. 1 part in 100 million. Computers can match that, but computers are macroscopic. How does a microscopic machine reach that level, and it turns out that there are a number of mechanisms for doing that. All right, so that's the setup. As I think everybody knows, the DNA polymerase is part of the central genetic machinery which generates all the proteins, that is, all the other machines. This is the machinery that generates other machines, the machinery for making machines. And it works, of course, by starting from a set of blueprints in effect, or a big pool of information, a hard drive, if you like, that's got all the genetic information, codes for making other machines, codes for making proteins. You got to copy that code every time the polymerase, every time the organism is going to reproduce, and that's the job of the DNA polymerase. It's the very first in this sequence of operations that the central genetic machinery carries out. The next one is to take one of the strands of the DNA and copy it into an RNA, messenger RNA, that's a temporary copy of the same information, or complementary information anyway. And then that messenger RNA gets used by the ribosome to generate finally a protein which folds up on its own to make a new machine. So that's the sort of the gist of what the central genetic machinery does. It's really powerful. It's a almost universal fabrication facility. You can make pretty much any machine, at least if it can be made at all out of organic molecules, using the central genetic machinery, and that's why life is so diverse and so powerful. That's why we can exist. All right, why use DNA? We'll go through this quickly because this is kind of a little bit of an aside. Versus RNA, right? There's two different kinds of nucleic acids, and they don't differ by very much. We'll talk about where the difference is down below. Probably a lot of people know it already. Why use DNA versus RNA? Well, it turns out that DNA is better for storage. It's more chemically stable, and that's its job. The DNA really doesn't do anything by itself. It's just a hard drive. It's just a passive pile of letters that should be stored fairly safely and safe from damage from the environment. By the way, the DNA does get damaged all the time. Every cell in our body, every gene in our body, every string of DNA in our body is constantly under attack, especially from oxygen, from oxidative metabolism. It's goobering up the letters in our DNA all the time. And so there are DNA polymerases whose specialty is correcting all that damage all the time. And without correcting that damage, you don't live very long. So really important. All right, but RNA, which is only slightly chemically different, is usually found in single-stranded form. There are exceptions to this, but usually only in single-stranded form. Looks like this when it's melted, but then it partially folds up and you get some double-stranded regions intermixed with single-stranded regions, and sometimes it folds up in quite complicated shapes. The ribosome itself, for example, is mostly RNA, whereas the DNA is usually long, perfect strings of double-stranded, double helices, better for storing information safely. This is much better. I mean, RNA is much easier to degrade chemically, which is what you want if you want a temporary copy, right? If this is a messenger RNA, and that messenger RNA is going to be transcribed by ribosomes, then you don't want it living too long because it's making proteins all the time. You want to be able to control that, so you want to be able to chew it up. So RNA is deliberately, in fact, less stable and easier to degrade than the DNA. All right. Here's the actual reaction we're looking at. Here's a piece of double-stranded DNA. Here's the template strand. That's the strand that's being copied. It's the strand that contains all the genetic information. We're going to bring in a nucleoside triphosphate, or nucleotide as they're called. That's what this thing is meant to represent. It's both a power source, these little three balls here, that's the triphosphate group. That provides the power that runs the machine, and it's also the building materials. The rest of it is the new base or the new residue that's being incorporated into this, what's this called? This is called the primer strand here, this one. So template strand here contains the genetic information. primer strand here is the one that is being copied and newly created by the DNA polymerase. And it's got to do it in a way, remember. It isn't good enough to just polymerize a random polymer, of course. It's got to do it in a way that every one of these letters gets read as the new bases are incorporated. All right. Here's the actual, we're going to need a little bit of truly atomic scale information and resolution and structure along the way here. Let's get a little bit of it right at the beginning. We'll talk a little bit more about it again later when it comes up. Here's the actual process. Here's the parent DNA. The helicase is moving this way, unwinding the DNA. There's a polymerase here and another polymerase there that are copying the two strands, one forward, one backwards. And then at these two junctures, here and here, these guys are the nucleoside triphosphates, or NTPs, that are being incorporated. I think everybody knows there's a base. That's the actual letter A. This is an A, yes, but there's also a T, a G, and a C, so 4 different letters. There's a sugar that's attached, that the base is attached to, and it's sort of thought of, it's sort of the center of the molecule. And then there's this triphosphate group on the other side of the sugar. And the reaction that we're going to be looking at is actually cleaving that triphosphate group right around here, meaning that the rest of this stuff goes away. And this phosphate is the thing that hooks the new base pair, if you like, into the growing DNA. Yeah, here's a way of representing that in a sort of cartoon format. Here's the energy, here's the triphosphate group, here's the sugar, and here's the letter or the base. Here's what that same molecule looks like in real life, rather than sort of as a diagram up above. This is the sugar. This is the triphosphate group. We'll see crystal structures of these later. Here's the base. You can see it isn't a flat molecule. It's sticking out in space and stuff. You can also see that there's a lot of single bonds in here. This whole triphosphate group can wiggle around quite a bit. There's also some wiggling that can go on in here, and that becomes relevant later on. We'll talk about that. Also, let me just point out at this point that there is a difference between DNA and RNA. When it's DNA, that little D there means deoxy for DNA. There's nothing here. There's a hydrogen here. There's no OH group there. It's deoxy because if you're thinking of RNA or ribose as the sugar, that there should be an oxygen, there should be an OH group there, and it's deoxy because that OH group is missing. On the other hand, if it were an RNA, then there would be an OH group here, and it turns out that while that difference is really minor, it makes considerable difference in the way RNA and DNA behave. And one of the big differences, I mean, there are a number of them, but one of the big differences is that that OH group is a little bit chemically reactive, and in a way that the RNA itself is susceptible to. And so that 2' OH group oftentimes participates in cutting up, in the RNA cutting itself up. And if you've got any divalent cations around, magnesium, calcium, that kind of thing, then together with the 2' OH group, RNA tends to cut itself to pieces, and that's good as far as the cell is concerned because, again, you only want a temporary copy of RNA usually. You want to be able to get rid of it easily. It also means that RNA is easier to cut up even if you're using an enzyme. All right, here's the overall reaction from a sort of chemical reaction point of view. A DNA with N base pairs, however many N may be, plus a new nucleotide is going to become a DNA with an extra base pair, N+1. The nucleotide's going to get incorporated. And we're ejecting this pyrophosphate. Again, the pyrophosphate is this last 2 phosphates. If this were normal ATP, this is deoxy ATP. The ATP that, or sorry, this is deoxy ATP. The ATP that you're used to using, or talking about, the thing that fuels every other machine in the cell, that's ribo ATP. So it does have the OH group here. And it gets cut here. Only the last phosphate gets cut off, and then you get ADP, right? Adenosine diphosphate. In this case, we're going to create an AMP, an adenosine monophosphate, but that's actually really not right, because this adenosine monophosphate is actually what gets clipped or attached or bonded to the growing DNA, so it never is free. It's always part of the DNA. And this diphosphate group is what gets ejected off. So that's what this PPI stands for. It's two phosphates together. Here's the reaction from a somewhat more structural diagrammatic point of view. At the end, here's the growing primer strand. This is the growing strand of the DNA. There's another strand over here. That's the template strand. And at the end of that growing strand is an OH group. It's called the 3' OH group. That's the big guy that's reactive enough so that a new nucleotide can be stapled in at that point. What that OH group is going to do, as we'll see in more detail later, it's going to attack the alpha phosphate here, the very first phosphate in the set of 3. And the result is going to be over here that a new bond gets created between the end of the primer strand and the nucleotide that came in, and now this nucleotide is part of the growing primer strand. And now the primer strand continues in that way. Again, there's a template strand out here, and the next thing that's going to happen is the next template letter is going to get read, and another NTP is going to get incorporated and grow the strand even longer. Yes, and then the pyrophosphate, or the diphosphate sometimes it's called, is what gets ejected. Okay, here's the actual structure of the polymerase. And I'll take some time talking about this because it's important that everybody have in their mind this structure. Otherwise, a lot of what I'm going to say afterwards isn't going to make any sense. First of all, here's the DNA. Here's the DNA double helix. So there's a strand here. These dark gray guys are one strand. And that's the primer strand. You can see if you follow the backbone up here to the end, it terminates there. That's the strand that has an end that's going to keep growing. And there's the 3' OH right there. That's the reactive end. The other strand that's kind of hidden behind, but it's back here, and it circles around and continues over here, that's the template strand, and it continues out here, right? Here's the template base that's being copied right at this moment. That's the transfer of genetic information from one strand to the other. That's the key event. A case could be made that it's the key event in all of life right there, that little kiss. And then here's another base on the template strand. Here's another base on the template strand. So these are the guys that are going to get copied next, and then after that, and so on. So that's the DNA, and then you can see the base pairs here, right? These are the already existing base pairs. And this guy right here, the yellow guy, is, of course, that's the NTP, the nucleoside triphosphate or nucleotide that's coming in. The yellow part here is the base. The sugar is kind of hidden back behind. And this red and orange guy that's so prominent, that's the triphosphate group right there. That's the group that provides the energy. And you can see, look how the polymerase has positioned things. We haven't even talked about what the polymerase is yet, but we'll get to that. Look how the polymerase has positioned things. It's set things up, and this is part of its job, part of its magic in a sense, so that reactive 3' OH group right there, not the best color. Let's see, let's pick this one. That reactive 3' OH group right there is right next to the alpha phosphate. That partially hidden orange ball there, that's the alpha phosphate, phosphorus actually. And all of this together is the alpha phosphate. And you can see it's right there. All it's got to do is form a bond right there. It's right position for this OH group to attack that phosphorus, displace the rest of it. This is the pyrophosphate, and form the new bond. And so right away we see that the polymerase sets things up really well right from the beginning. That's part of the bonding process or binding process by which the polymerase binds to the new incoming nucleotide and sets it up for the for incorporation. Notice also that these two guys are hydrogen bonded to each other. They're already annealed to each other. The hydrogen bonds between this template base and this incoming new base are already made, and not only that, but this NTP is kind of sitting right where it's a little bit off from where it's going to be finally when it's actually part of the DNA. It shifts a little bit during the reaction, but it's close. It's already almost in the exact position where it will be. It's kind of almost already part of the DNA, right? Except for the fact that this bond needs to be formed. It's already in the right place. All right, the rest of it now. So that's the DNA and the nucleotides and all. What's the polymerase? Well, it's got 3 domains, 3 major regions. I think I'll get rid of all this now. And those are, I guess green is appropriate here, the thumb. So that's all of this. Yeah, and it's called the thumb. Because it's called the thumb, and this is called the palm, and this is called the fingers, because we think of this, of the shape of the polymerase as being a right hand. So this is the thumb coming around. The thumbnail would be here. And here's the palm, that's the back of the hand, right? And then here's the fingers coming down. That's what the analogy is. So thumb, palm, and fingers describes the basic geometry. And the thumb is lying, so this is the major groove of the DNA. This is the minor groove of the DNA. And so you can see that the thumb is sitting right in the minor groove. And if you look at the crystal structure at the atomic resolution scale, you can see there's all kinds of contacts between the thumb here and the minor groove. It's binding to the minor groove. A large part of the ability of the polymerase to bind to the DNA at all is in the thumb. And there are cases where the thumb starts moving around and helps the polymerase move, for example, along the DNA. Then there's the palm, and the palm doesn't play kind of as dynamic a role as the thumb does. It also helps the polymerase stick to the DNA. But its major role, its most important thing, are these 2 magnesiums right here, those 2 pink guys. Those are magnesium 2+. They're doubly plus charged. And there's 2 of them right next to each other. Why is that? Well, we'll see when we start talking about the catalytic mechanism, but especially this one down here is going to catalyze and make it much easier, reduce the energy of the transition state, reduce the activation barrier for this 3'OH to attack that alpha phosphate. So it's absolutely crucial to the whole mechanism by which the catalysis takes place. The other magnesium is also important, but it's more important because since it's positively charged, each of these oxygens, most of them anyway, all but a couple, are negatively charged. This triphosphate group has 4 negative charges on it. And so those 4 negative charges are obviously going to interact very strongly with the magnesium, especially in this environment, because most of the water has been excluded from this environment, so there isn't any competing water. So that magnesium is important for positioning and binding the triphosphate group, and the other magnesium is important for actually catalyzing the reaction, the attack of the 3'OH on the phosphate. Okay, so thumb, palm, and then finally, the last, and in some ways the most important, well, maybe not the most important, but equally important to pretty much anything, and the most dynamic part of the molecule is the fingers here, this blue thing. It just looks like a blue blob in this presentation. You really can't see it very well. But I'll draw things to help you see it. This little thing right here, you can kind of see that it's sticking out over these two, this nascent base pair here, this base pair that's about to be. And in fact, that moves. That thing right there is a movable hatch. It can open and close. When it's in the open position, it's up here somewhere. When it's open, nucleotides can come in. When it's closed, as it is here, it's in the closed position here, right? This is the closed catalytic complex. So when you take a crystal structure, you can only take a snapshot of one particular state of the machine. This state is in many ways the most important one. It's the state that's just about to do catalysis, that's just about to form the bond. Closed means that these fingers are bent down right on top of the template base and the NTP, just like it's shown here, and they're pinning it in place. They're holding it in place, they're holding it steady, they're holding it firm. They're also recognizing that this guy, the actual NTP, is arranged or oriented in the right way. If you get an NTP in here in some funny way, the fingers can't close, and then the catalysis can't take place. That's a major part of the way in which the polymerase chooses the correct nucleotide. We'll talk about that later. Anyway, it can open up, and when it's open, then new nucleotides can come in. So during the course of a cycle, a cycle starts with the fingers open, then they close, and after they close and basically check to see if the correct nucleotide is in place, then catalysis takes place, and then all kinds of things happen after that. We'll talk about that. All right, that's the structure. Remember the thumb, palm, fingers. Remember that the thumb is bound in the minor groove. Remember that the palm has got those two catalytic magnesiums, and remember that the fingers is the hatch that opens and closes to clamp down and form the tight catalytic complex. All right, that's the way. This is actually HIVRT. And, oh, gosh, can't spell. The RT stands for reverse transcriptase. It's not actually a proper DNA polymerase, though it can function as a DNA polymerase. And we'll see that it doesn't matter that it's kind of a funny type of polymerase because it's very similar to all the others. This is the polymerase that copies the genome of the AIDS virus. The AIDS virus has an RNA genome, so the reason it's called a reverse transcriptase is because it actually copies an RNA into a DNA second strand. It forms an RNA-DNA hybrid. We won't go into that too much, but suffice to say that it's a little bit of a funny polymerase, but it works the same way, except for minor details. It's essentially representative of all other DNA polymerases. That's what I use it. That's why I used it, also because I have a lot of good graphics for this particular case. Here's some others. So these are other DNA polymerases. This one is a bacteriophage polymerase. In other words, it's from a phage, it's from a virus. It's the replicase for that virus, but you can see it's got the same structure. It's about as far removed phylogenetically from us as it's possible to be. Viruses aren't even alive, but it's got a thumb, it's got a palm in light blue here, or cyan, and it's got fingers in orange here. All these pictures, the fingers are orange, the palm is blue, and the thumb is green. And it works by essentially the same mechanism. You can see that the shapes are different, but the structure is basically the same. You can see that the nucleotide, and right there is the base that's being copied. They're in the same position. I didn't draw in the 3' OH, but it's right there. And there's the two catalytic magnesiums. It's obviously doing pretty much exactly what this one up here does. And here is the one that's up there. You can see in the same color scheme. And of course, thumb, palm, fingers, catalytic magnesiums, and so on. Here is a repair enzyme. So this isn't a replicase. It's not in charge of replicating the genome. It's in charge of repairing mistakes or trouble or lesions, as they're called in the genome, in the DNA. And so it's simpler for that reason. That's why I put it in here. The actual replicases for eukaryotes are really complicated, and needlessly complicated for our purposes. There's the thumb, there's the palm, there's the fingers. It works the same way. It's simpler. Its fidelity is low because it doesn't need to be high for a repair enzyme. It only works on a few bases at a time, and mistakes aren't so important for this one. Speed and other things are more important. But you could see same structure. Here's one from an archaebacterium. So you know there's eubacteria and archaebacteria. This is Thermus aquaticus. This is the thing that's used in PCR widely. But again, thumb, palm, fingers, works the same way. The fingers are still a hatch. The sort of nascent base pair is there. The triphosphate is positioned. The two catalytic magnesiums, everything. And then finally T7, this is another phage polymerase, but it's very similar to Pol-1 from E. coli, for example. So thumb, palm, fingers, and so on. So when we talk about any one of these polymerases, we're basically talking about all of them. There are variations in structure, there are variations in purpose, there are variations in fidelity, there are variations in mechanism, but they all work very roughly in the same way, and they all look the same way. All right, let's see what it does now. And this is just for, this movie doesn't teach us a ton, but it gives you a visual impression, which is useful for what we're going to do next. This program doesn't play movies, so we're going to switch to PowerPoint here. Here's the movie. And, oh, let me stop it for a second. Notice, here's the genetic letters of the template strand. So this is the template strand. That's the thing that's getting copied. New nucleotides are coming in, but this movie doesn't show those nucleotides. That's the fuel and also the building material for the new strand. You can see it's double-stranded down here, and so somewhere buried up in here, actually, let's run it back a little bit. Let's see if I can run it back. There we go. Right in here is where the replication is taking place, where the new bases are being incorporated. And let's see if I can, don't know how to erase. So I shouldn't draw too many marks, I guess, after this. There's the fingers in yellow. The yellow thing is the hatch. You'll see it opening and closing. The rest of it, this is actually T7 DNA polymerase. It's one of the ones I showed up above, but it's not the pretty picture I showed earlier. And we're going to see two different versions of actually 3 runs of 2 different versions of this movie. First 3 runs show sort of a cartoon of how it works. The second 3 runs show a much more realistic version of how it works. All right, let's let it just run. Go. There we go. So you can see the fingers opening and closing like a staple gun. Every time it closes down, a new base gets incorporated, and there it's starting over again, and it's just chewing right along, running along the template strand, making a double-stranded DNA that it's moving along. And there it goes again. And if it's doing its job right, every single letter that goes in matches the... All right, now here's the more realistic version that has thermal fluctuation. So now everything is getting buffeted by solvent, there's all kinds of energy, right, in the surroundings that are bouncing and pulverizing into this thing. So it doesn't work like that nice, clean thing that you saw. The fingers are still opening and closing. On average, they're doing the same thing as what we saw before. But statistically, they're wiggling around and fluctuating around like crazy. And it's good to keep in mind that this is what's really going on with molecular machines. All the movies you see don't show these fluctuations usually. I've never run across one, actually, that does show them because it makes things hard to see. The earlier movie is much easier to understand, right? This one's hard, it's hard to see what the thing is doing because it's shaking so much. But those shakings are... this is not imaginary. This was a real calculation, and that the fluctuations that you're seeing are typical, are the right order of magnitude for what's really going on at the molecular scale. All right, back to the notes. Here's just another view that's helpful when we go a little further to get a feeling for what's going on. In fact, this is the same polymerase that we just saw the movie of. This is T7. Mostly we're going to look at HIV-RT because that's what I have the best graphics for, but this is T7. Again, another viral polymerase, but it doesn't matter that much. And here's a sort of cartoon view that shows functionally what's going on. First of all, this brown thing here, that's the body of the polymerase. And it shows it fitting into these grooves on the DNA, into the backbone of the DNA. And you could see in this movie up here that it seemed to sort of spiral around the DNA and sort of like a nut on a bolt, right, on a threaded bolt. And that's exactly what happens. If you look at the structure of the actual DNA, you can see that there are thread lines, there are grooves in effect, or interaction lines, curves, on the interior surface of the polymerase that follow the backbone of the DNA. And so it really is just twirling along on the DNA like a bolt on a nut, powered by the nucleotides that are coming in. And, oh right, here's the fingers, yellow again, just like in the movie, that can open and close. There's a little spring, there's a hinge, and there really is a hinge, and there really is a spring in the molecule. It's kind of hard to see them sometimes, but they're there. And so this can open and close, and that hatch or fingers domain interacts with the polymer, with the incoming nucleotide here. Here's a top-down view where the top stuff, all this stuff's been cut away, so you can see what's going on inside, so to speak. Here is the template base that's being copied. Here's the nucleotide. There's the triphosphate, right, the 3 dots, the 3 phosphates that's coming in right now. And these little red zones that you see in the picture, those are areas of interaction, lots of interaction right in the nucleotide, as you can imagine. We'll talk a little bit more about that. And in this particular polymerase, there's also some interaction between the lid here, the fingers, and the thumb here. This is really the thumb coming around. And that's not true for all polymerases. It's not true for HIV-RT, for example, and it's not true for many other polymerases either. But something like this is what's going on all the time. And in fact, these sticking interactions, basically this hatch sticking to all this part in here, that's a key part of how the machine works. The spring is also a key part. We'll talk about that as we get to it. Let's see, what else should I say here? I guess the only thing to say is to point out again, you can see really well right now. This is the template base that's being copied. That's the nucleotide coming in. And you can see they're sitting right on top of the DNA. This is the DNA underneath. They're sitting right on top of the DNA, almost exactly as they would if they were both, if this nucleotide were already part of the DNA. And that's part, of course, how the machine works. All right, finally, that's all sort of background and structure. Now let's look at the kinetic mechanism. So here's where kinetics come in. We haven't even talked about thermodynamics yet, but we need this kinetic mechanism to sort of organize the discussion from this point out. These are obviously cartoons, not very good ones, in fact, but they get the idea across, I hope. So there's 5 steps. 1, 2, 3, 4, 5. All of them are quote-unquote elementary steps, and we'll talk about what that means in this case, because they aren't elementary in the sense that we've talked about in the rest of the class. The very first state of the machine is with no nucleotide present. There's a gap there, right? The fingers are open to admit a new nucleotide. There's the palm. The 2 magnesiums are there, but they're not shown in this picture. Here's the thumb wrapping around the minor groove. And the first thing that happens is that a new nucleotide comes in and sticks itself into the correct position. And so the nucleotide binds at that point. This is a reversible mechanism, which means the nucleotide can come off at that point. In fact, that's important because You don't want every nucleotide that comes in to stick really hard because it might be the wrong nucleotide. And you want to check first whether that nucleotide is the right one or not. And that happens mostly at the fingers closing step. So you've got to have the possibility of this coming in, not binding very well, binding weakly, and then coming back off. So this is a reversible step. Notice that this first thing right here looks just like the beginning part of the Michaelis-Menten mechanism, right? In Michaelis-Menten, it would be E plus S, K1, K minus 1 goes to ES, and that's exactly what's happening here. So at least that first step is very consistent with Michaelis-Menten. Of course, there's 5 steps here, so it can't really be Michaelis-Menten, or at least not the simple mechanism. But at least the first part looks like it, and we'll see that that's significant because in fact, this enzyme is a Michaelis-Menten enzyme as far as its behavior is concerned, at least. I said earlier that there's lots of enzymes that aren't Michaelis-Menten but behave like Michaelis-Menten, and this is one of them. All right, so now the nucleotide is binding, and of course, as I've emphasized earlier, this is the place where the nucleotide is reading the template, reading this template base, and therefore the place where the genetic information is copied, and we'll talk a lot more about that later. Then the fingers close, and this is a slow step. We'll talk about why it's slow. There's a functional reason why it's slow, and there's also a physical reason why it's slow. There's a sort of physical way of understanding it, but also it should be slow. We'll see that it's important that it be slow because this is, like I said, part of the testing process to make sure it's the correct nucleotide. And that slowness is necessary to do the testing well. So the fingers closed, this is a slow step. In fact, it's slow enough compared to all the other steps in the whole process. All the rest of them are pretty fast compared to this one. At least 4 times faster, and a lot of times 20 or 30 times faster. And so this is also the rate limiting step. And because all these other steps that come afterwards are much faster than that rate limiting step, they hardly contribute to the overall turnover rate at all. In other words, if we neglected all these other steps and just said that the K for this step was K-cat in the Michaelis-Menten mechanism, then you can see this first part here would be a Michaelis-Menten mechanism, and that's in fact the way the thing behaves. All right. The next step is actual catalysis. So we've now formed, the fingers have closed, we've now formed the catalytic complex. This is exactly the thing we were looking at in the pretty picture above. Catalysis takes place. We'll talk about what that catalysis is. Some heat is released, and so this is a thermodynamic driving step, and it's important to point that out. Actually, the binding up here is also thermodynamic driving steps. Every one of these steps has a negative delta G associated with it. In other words, they all go downhill in energy. They are all thermodynamically favorable to go. But this is one of the most notable ones down here. However, it's a pretty weak thermodynamic driver. The delta G is only about 3 kilocals per mole here, and so it's not a big, strong driver. Still, it is one, and we'll talk about the mechanism by which this catalysis actually happens. I've already talked a bit about it, but we'll talk about it more when we get to it. The next thing that happens is, all right, now after catalysis, the nucleotide is actually hooked into, covalently bonded to the growing primer strand here, right? But the pyrophosphate is still there. The 2 pink dots right there, that's the pyrophosphate. It's still there. You got to get rid of it in order to get back here where you want to restart the cycle. So there's PPI release. That's also a thermodynamic driver because entropically, of course, releasing a product, especially a product in low concentration in the cell, which PPI is favorable, and so this is also it's really part of the overall delta G of the reaction. In fact, the overall delta G of the reaction is sort of partitioned amongst all these steps, and so there's a little bit of it here, 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 and so on, even a little bit of it over here. It's distributed. That's why the machine works, of course, right? You need something to power each step. Anyway, so the PPI is released, and now we're ready for maybe the most dramatic part, well, one of the more dramatic parts in this, And that is that the polymerase has to move to the next site, right? Right now, it's still stuck at the same site it was at. It hasn't moved, and so it can't take in a new nucleotide. The fingers are still closed. It can't take in a new nucleotide for that reason either. So the fingers have to open, and the polymerase has to move. No one's quite sure where the polymerase moves. It could move here. It could move at other places, but it's probably here. This is the most likely site for the translocation step. There have been simulations done that show, well, seem to show anyway, that when the fingers open, it actually pushes the polymerase forward. That's called a power stroke. We'll talk about that when we get to it. All right, so that's the mechanism. After you've opened the fingers and moved the DNA, you're back in the original state with open fingers ready to accept a new nucleotide, and the DNA in the correct position, the DNA polymerase in the correct position on the DNA in order to stack a new nucleotide on there. That's the mechanism. I mentioned up above that this step, for example, is considered to be elementary. So are all these others, but in fact they're not single, at the atomic scale, they're not single steps. And so you can think of it in the following way. If I have a really simple reaction like this, two hydrogen atoms coming together to form a hydrogen molecule, then you've got a simple, clean a reaction energy diagram like this with a single clean kinetic barrier hill, right? If you've got something like what we've got here, you can think of it more like this. There's actually a whole bunch of steps that are truly elementary, but they're all very fast. It's a rugged top. It's an energy hill that's got a whole lot of corrugation and jumps on it. But since it happens fast enough so that it's much faster than anything that comes before or after, we can still treat it as an elementary step. And I emphasize this here because that's common in biochemical situations. Almost all the elementary steps in biochemical kinetic mechanisms aren't truly elementary. This I call just elementary straight. Here I put elementary in quotes because it acts elementary for our purposes, for our purposes, but it isn't truly elementary in the sense that we talked about earlier. All right. We've looked at this crystal structure already, but it doesn't show everything we want to show, so I'm going to represent it from now on with this cartoon. And you can just see that I traced this cartoon from this picture, in fact, but now I can change the cartoon, right, to show different states of the polymerase. So there's the fingers, there's the fingers, there's the palm, there's the palm, there's the thumb, there's the thumb, there's the DNA going through the middle here. Here's the template base that's being copied, and there it is in the same color in blue. Here's the new nucleotide coming in, and there it is. And notice there's the triphosphate group. There's the two key magnesiums right there. And there's the crucial 3' OH group, reactive 3' OH group that's coming in. Yeah, so that triphosphate group is this guy, right? It's going to get cleaved right here. The pyrophosphate's going to go off, and this phosphate's going to become part of the backbone in the DNA. And then, yes, there is the template strand that's continuing out. That's this part right here. And so, okay, that's the picture I'm going to use from now on. All right, so let's talk about the individual steps. Let's see what kinds of things are driving it if we can. First of all, there's the NTP binding step. The very first step in the mechanism we just went through. That's the process where a new nucleotide comes in and sticks itself, the fingers are open, you notice, sticks itself into the gap that's present because the polymerase has already moved up, right? The base pair below was just polymerized a minute ago, say, but now the polymerase has moved up and left a gap. The nucleotide can stick in there. The delta G associated with that binding step by itself, right? It's not completely bound yet because the fingers are still open. When the fingers clamp down, it's really bound, but it's in an earlier stage right now. Even so, as long as this base, this template base, is in the correct position, it's not hanging out here someplace. In some crystal structures it is, so it can be in the wrong place. But if it's in the right place, then there's a negative delta G. So if it's unstacked, if this blue guy is in the wrong place, the delta G is unfavorable. The nucleotide can't bind. But once the polymerase, once the template base is in the right place, then it can hydrogen bond, of course, with the new nucleotide coming in, and all the other interactions are good, in fact. And then the delta G is negative, but it's not huge. -4 kilojoules per mole, a pretty weak interaction, or a pretty weak delta G. What kinds of things are going on? Well, there's all kinds of interactions, of course, that are taking place in this case. Let's look at some of them. First of all, of course, there's the hydrogen bonding to the template base. So here's the template base. Here's the new base coming in. This is, I forget, I think this guy is an A, and that must mean this is a T, so it's just got 2 hydrogen bonds. If it were GC, it would have 3. But, and the GCs are, you know, a little bit, have a little bit stronger hydrogen bonding, because it's 3 versus 2. But anyway, that's certainly happening, and that's going to be, I put a delta G here as negative or positive. Negative meaning the delta G is negative, which means it's a favorable thing. It's going to promote binding, or it's going to help with binding. And this is one of those, right? This is a favorable thing. It's going to help stick the NTP into the polymerase. Next thing is interactions between the magnesiums, the catalytic magnesiums, and the phosphate group, right? So that's here. Let's go back up here. Right there, right there's the phosphate groups. Let's zoom in here. There's the magnesium, sorry, and this is the phosphate, the triphosphate. You can see the triphosphate is negative. The magnesiums are positive. These got to be sticking hard, right? So there's very favorable interactions there. On the other hand, remember with electrostatics that in order to create this situation, you've got to strip water off. There's the desolvation energy, which is oftentimes almost as big as the new favorable interactions energies, or even can be bigger than the favorable interaction energies. So exactly how much net delta G there is not so clear. I haven't put numbers on these things because they're not known. But it could be that this is not that favorable. I've marked it as favorable because it probably is. But that's another kind of interaction. This interaction is also very important later when, well, we'll talk about that, when the pyrophosphate is going to be released, and also when the fingers come down to close. Then the triphosphate desolvation, right, that's another contribution, an unfavorable one. Magnesium 2+ desolvation, also unfavorable. There's stacking, right, between, yeah, okay, we can look at it up here. Between this guy and the bases below. There's also stacking between this guy and the bases below. And those are favorable, of course. Those stacking interactions are the main stabilizing energy for the double-stranded DNA itself on its own, and they're favorable here too. And then let's see, so that's this interaction, right? This interaction right there. Then there is NTP, internal rotation energy. Remember I pointed out all those bonds. We'll look at them again later. In the nucleoside triphosphate that can rotate. Well, once they're bound in this tight complex, they can't rotate anymore. So the whole nucleotide loses a bunch of rotational entropy when it binds, and that's unfavorable. If there are bound waters in that pocket, and I think there are, I'm quite sure, in fact, that there are, then those are going to get released when all of this happens. That's a favorable water release entropy contribution. The, let's see, template base backbone entropy. Oh, yeah. So the template base, the template strand is coming off over here. When this binds, it may be that starts restricting some of the movements possible to the template base, in which case that would be an unfavorable contribution. And then there's the template base stacking itself, which would be a favorable contribution. All right, I've just listed these. I'm not sure these are all of them, but they're probably most of them. I'm not sure all of them I've got the sign right necessarily, because most of these things are not known. But in general, these are the kinds of things that you're looking for if you want to understand why this event occurs, why the binding process or event happens at all. All right, that's the binding step. Now the fingers closing step. Maybe the most important step in the mechanism, though there are also, of course, other important steps. So in this case, the fingers clamp down. I've already said that this is slow and rate limiting. That's why this all behaves according to Michaelis-Menten. There's a spring. The hinge is around here on this picture. And it takes energy to bend that hinge because the natural state for the fingers is to be open, the way it is drawn here, in fact. And in order to close it, you've got to bring it down, and that's important. We'll talk about that. That's basically a kinetic barrier for this step. It's part of the reason why it's slow. You got to bend that spring. You got to get energy from the environment in order to bend that spring. You got to reduce the entropy of the environment, of the surroundings, in order to bend that spring. Once it bends, though, and comes in contact with this nascent base pair, the NTP and the new primer base, then there are quite strong interactions between the tip of the fingers and the base pair itself. And some of those are hydrophobic residues. Every single polymerase I've looked at, and I've looked at a couple dozen, have hydrophobic residues right underneath here. And they're there, obviously, to stack on these relatively hydrophobic bases. Some polymerases have actual aromatic residues there, like phenylalanine or tryptophan, so that they stack almost exactly on top of these guys as if they were already another DNA base pair. So pretty strong sticking going on right there. And then there are always positively charged residues, lysines and the like, on the this side of the fingers as well, and they come in and make presumably favorable contacts with that negatively charged triphosphate group on the NTP. So those are the main interactions going on. This hinge bending is unfavorable. These kinds of sticking interactions are favorable. The overall delta G is around 16 kilojoules per mole if there's an NTP in the pocket. So suppose the fingers tried to close when the NTP is not there. Suppose that's gone. Then what happens? Well, then the delta G estimated, these are crude estimates, but roughly is about plus 11 kilojoules per mole. In other words, this whole process is only favorable because the fingers can stick to this NTP, and it wouldn't happen at all, or at least it could only happen against thermodynamics, very rare, very tough, if that NTP were absent. Also, let's suppose the NTP were present, but it's the wrong NTP, not the one that matches the template over here. And so in that case, it sticks in here kind of funny, right? It tries to make a base pair with this, but it can't make it very well. Gets in there in a funny orientation, and then what happens? Well, all these interactions don't work so well, right? That's part of the way in which the closing of the fingers tests for whether that's the right NTP or not. Yeah. Also, from the point of view of controlling the cycle of events that's happening, the fingers can't close until there's an NTP there. And so you can't get the fingers closing before the binding occurs. The fingers closing pretty much has to happen after the binding occurs. What are the contributions in this case? Well, there's the hinge strain energy. I've talked about that. Then there's these nonpolar stacking interactions here with the NTP base and the template base. With both, it's actually, if you look at the crystal structures, there's stacking interactions with the NTP base, but also with the template base. It isn't just for the NTP base. There's the charge-charge interactions. I already talked about that between the fingers and the triphosphate. Probably there's some water release. That's probably favorable. All these guys get negatives on or minus signs on the delta G here because they're all favorable. And then probably when the fingers close, the remaining template strand here gets a little bit restrained. I don't know this for sure, but I suspect that when the fingers close, it restrains some of the backbone chain entropy in the remaining part of the template strand. So that would be an unfavorable contribution. Anyway, again, these are not all the contributions. I don't know that they're all even correct, or that they're all even major. These are pretty much my surmises, but they give you some feeling for why the fingers would close in the first place. Yeah, here this just shows, this is a blow up of one shot from the picture above that shows, yeah, the interactions going on, especially in here between the fingers, which is kind of sticking out over the top. You can't see it in 3D here, but it's sticking out over the top of these guys and interacting favorably here and here and here. There's also, of course, stacking interactions that are taking place between these guys and the bases below. All right, now the third step, the catalytic step. This is the step where the actual bond forming and bond breaking reactions take place. What's going to happen? We talked about this a little bit before. Here's the NTP coming in. It's got its triphosphate group on it. Here's the sugar. Here's the base. I've just reduced the base to a little square here, and there's the 3'OH, the reactive growing end. This is the primer strand. The rest of the primer strand going this way, actually. Probably better to draw the arrow this way. It's coming up from above. This is hooked to the next residue down. There's the base. There's the sugar. There's the 3'OH. It's positioned by the polymerase to attack that phosphorus atom and form a new bond. Again, yeah, I said this before, it's a thermodynamic driver, but the delta G is pretty weak. This will go, and it will go just fine thermodynamically, and yes, it will be favorable mainly because it's releasing heat at this point. It's not an entropic effect, I don't think anyway, but it's a relatively small one. And again, just to emphasize, I've said most of this before, but here's the two magnesiums that are positioned to hold the triphosphate here, and also right next to the 3'OH group. And all the 3'OH group has to do is form a new chemical bond right there. It's about, if I remember right, it's about 3 or 4 angstroms away, 3.5 angstroms away. It's farther away than it's too far away to form a chemical bond on its own. So these things have to move a little bit, but it's very close. And there, yeah, this is the alpha phosphorus right there, and this whole thing is the alpha phosphate. All right, here's the actual mechanism. We won't spend too much time on this because this is now getting into the details and not really important for how the whole thing functions as a machine, but it's useful to see what the polymerase is now doing right at the catalytic step. So first of all, this right here is the primer strand. And, this is the template strand, and this guy is the primer strand down here. So this is P, and this is T. And this is the template base right there. That's the thing that's being copied. Here's the new NTP coming in. Let me blow it up so you can see better. Here's the two magnesiums, of course. There's the 3'OH. That's going to attack right there to get that phosphorus and form the new bond. What good does those magnesiums do? Why is this magnesium, this A magnesium there at all? Here's why. It's because as part of the mechanism by which this 3'OH attacks that phosphorus, you've got to lose the hydrogen from the OH group. Well, OH groups are not good acids. They are very poor acids normally, right? But with this 2+ charge here, the formation of that minus charge on the oxygen is much easier, right? As soon as you lose the hydrogen, you get strong interactions here. That's a favorable contribution to the delta G. Therefore, the pKa of this group, the ability of that group to lose its hydrogen, is much higher in the presence of this magnesium than it would be otherwise. And now that's a reactive group. That O-, that ionized OH group is reactive and much more likely now to be able to attack there. So this is the stabilization of the transition state. This is the actual truly elementary step where the transition state is stabilized and where the reaction is speeded up enormously by the catalyst, and this this magnesium 2+ is one of the major parts of that happening. All right, to look at the next steps, this one is the next step. Yes, now that 3'O- attacks the phosphorus, electrons move around, and that's obviously these are small, truly elementary steps now. But notice that all these steps are part of what I'm calling a single elementary step, namely this one. And so this is another example of what I talked about above, that in biochemistry, quote-unquote elementary is different from truly elementary. These guys here are truly elementary. That the whole step as a unit is not truly elementary. All right. Last thing then is the pyrophosphate goes away, the new bond forms here, and now this guy is just a new base added onto the end of the primer strand, a new residue added onto the end of the primer strand. A couple more diagrams here just to fill that out. Here's the positioning in the structure itself. Here's the double-stranded DNA coming up this way. This is the primer strand. And here's the new nucleotide coming in. There's the alpha phosphate right there. Here's the two magnesiums. You can see that this magnesium is making all kinds of contacts with the negatively charged oxygens on the triphosphate group. And you can see that this magnesium right here is right close to the 3'OH group. And you can see there's a pretty good gap here, too big for a normal bond, but that will close up. Positioned well, anyway, to attack that phosphorus. And let's see, what's this? The same thing, I think? Yeah, except it shows the template strand up here also. And I think that's pretty close to what we've already seen, so I won't make any more comments on that. All right, so that's the catalytic step, the actual bond breaking and formation. What's the next step? The next step, this is the fourth step in the 5-step mechanism, pyrophosphate release. That's pretty straightforward. The fingers are still closed, but notice if we look up here, for example, and get rid of some of this extra pink stuff here, that this is the pyrophosphate right here. And notice, and this is in the closed. This is the closed clamped arrangement. It's still. I mean, if you were to draw all the atoms here, this would look a little bit more buried than it does here, but it's not very buried. It's possible for this thing to get out without the fingers opening. In some polymerases, I have no doubt that the fingers open first and then the pyrophosphate releases, and it doesn't really matter which order it happens in. But for simplicity and concreteness, I have said that the pyrophosphate releases first. I think that's true in most cases, or thought to be true in most cases anyway. So the pyrophosphate lets go. Before, look, down on these cartoons, there's the new base pair, right? That used to be the primer, the template base on this side, and the NTP on this side, but now the pyrophosphate's been cut loose, and now that it's cut loose, no more bonds holding it in up here. These magnesiums are no longer strong enough to hold it in place, so it comes out. And now you've got less interaction with the fingers, right? The fingers before had favorable interactions with that pyrophosphate, and now those interactions are gone. It still has the interactions with the top of the base pair, but it's lost some of its interactions. The delta G for pyrophosphate release, again, this is my estimate, at about -8 kilojoules per mole. It depends on how much pyrophosphate there is out in solution, right? That's the way delta G works. And so this number isn't hard and fast. It depends on pyrophosphate concentration. But I've chosen it for a typical value inside bacterial cells where the pyrophosphate concentration is about 0.5 millimolar. Yeah, so notice that this delta G is 2.5 times, more than 2.5 times bigger than for the bond breaking and formation steps. So this is a bigger thermodynamic driver than the actual catalysis is. And just very, very briefly, the things that contribute to it is, first of all, the delta, the entropy change for the release of a low concentration product, the unfavorable delta H for breaking all these electrostatic interactions in here, the favorable, probably, yes, almost certainly, solvation of this guy by waters and any counterions that are in solution, right? There's going to be some sodium out here maybe, or maybe some magnesium for that matter. That, you know, and this can even come off with the magnesium. It can take a magnesium with it, right, if it wants to. That means breaking the bonds of the magnesium to the polymerase, but okay, it can happen. Anyway, if there's some magnesium in solution, and there has to be, polymerases don't work without magnesium, so in solution. So these magnesiums are exchanging with magnesium free in solution all the time. So anyway, counterions out here, all that is resolvation, right? So that's the delta H of solvation. Also, there's a delta S for the rebinding of water or maybe release of water. It's hard to know what the sign of this is, but that's another probably contribution. Next step, and last one, the translocation and fingers reopening step. Again, I'm not sure that translocation happens here, nobody is, but this is probably the most likely place. Once the PPI is released, having lost those nice interactions between the fingers and the pyrophosphate, the fingers being closed is now unfavorable. This spring is under strain, and now it's not stuck closed anymore. It pops open, springs open, and at more or less the same time, the polymerase moves up by one step along the DNA. How does it move? What drives it to move? That's not really known. We're not even sure where it happens, and it may happen in different places, at different stages along the mechanism in different polymerases. It isn't necessary that all polymerases be exactly the same on this point. And so here are some possibilities. First of all, it can be what's called a power stroke. So this is from the molecular machines field, which is my field. A power stroke is where there's some actual physical push or force that pushes the DNA along, the polymerase along the DNA. And there's some evidence for that. There are simulations, for example, that show that when the fingers spring open, there's a lever called the O helix that moves in a way that pushes, basically when the fingers open here, there's a little lever back here that pushes on the base from behind and pushes the polymerase forward. Simulations aren't the best evidence. They're better than nothing, but they aren't the best evidence. So whether that's really true or whether it's true for all polymerases, even if it's true for some. I mean, this O helix that I mentioned doesn't exist in HIV-RT, for example. So whether it's true for HIV-RT, even if it's true for the polymerases on which these simulations were done, we don't know. But that's a possibility. Another possibility is something called a fluctuation ratchet. In that case, all that happens when the fingers spring open is that the polymerase gets, and the thumb moves too, crystal structures show. There are small changes in the palm too, but mostly in the thumb and fingers. Then the polymerase gets loose on the DNA. It can start diffusing or fluctuating up and down on the DNA. It starts rotating on the DNA all by itself until it reaches high enough, it gets high enough up so that this next this one right here, the next template base, the next thing that's going to get copied, stacks. So if that folds into place, let's show, yeah, like this one is, right? This one's stacked on top of the lower bases. Well, that can come out. There are crystal structures that show this guy out, and it's in a funny cocked position, and it can probably flail around all over the place. But once the polymerase moves up far enough to allow it to come in, then it's favorable for it to stack, right? And then now it's ready to bind the next nucleotide. And so one mechanism is that the polymerase fluctuates up and down on the DNA until it stacks, and then it gets locked in place. That's why it's called a ratchet, right? You can go forward up to a point, and then things lock in place, and then you can't go back. That's why a ratchet always goes forward. So this is called a fluctuation ratchet mechanism. Again, it's one of the standard mechanisms in the molecular machines and motors field. So those are two possibilities. No one knows for sure which one is actually used. Probably there's somewhat better evidence for this first one, but that's weak evidence, so we don't really know, and like I say, it may not be the same for every polymerase. The polymerase generates force. It has to generate force. It has to winch the DNA in. If you're thinking of the polymerase sitting still, then it has to pull the DNA in, or if you think of the polymerase as moving and the DNA sitting still, then it's got to pull itself forward along the DNA. And so there are forces generated. What's the nature of those forces? How big are they? What generates them? Well, we don't know all that much, though there have been a bunch of single molecule experiments designed to eliminate that or to investigate that. This is a particularly difficult system for single molecule force measuring experiments to work on, and so the results are mixed and unclear at the moment, but I can give you sort of the gist of what's been found so far. So first of all, how much force could the DNA polymerase in principle generate, even if it used all? So the overall delta G for the reaction for the whole cycle, the whole 5-step cycle, is probably around minus 25 kilojoules per mole. Even that number is quite uncertain. This is typical of biophysics, actually, biophysical chemistry, physical biochemistry. They're all numbers like this, really fundamental numbers, like even what's the delta G for the overall reaction? You would think that would be known. This thing has been studied for many decades now, 50 years probably, and we still aren't certain what that number is. It's just hard to measure. But this is a decent estimate, and it's a fairly recent one, like 2016, I think. And so let's say that's the number. Then that is the amount of energy, the delta G, that can be put in to the process of doing work by the enzyme, by the polymerase. The force that the polymerase generates times the distance through which the polymerase moves has to equal the total amount of delta G that's put into that step. I'm saying here that it's all the delta G for the whole thing. That can't really be true because delta G has to be put into other steps. But let's suppose that we would put it all into that one force generating step. If that were the case, then the maximum amount of force that the polymerase could generate would be the delta G available, 25 kilojoules, divided by the distance that it moves, and it moves 3.4 angstroms. That's the distance along the DNA, between base pairs along the DNA. That's a well-known number. It doesn't vary very much. So this one we actually do know. And that would give us 1.2 times 10 to the minus 10 newtons. That's the MKS unit of force. Chemists are unfamiliar with force units oftentimes, so I point that out. And in the single molecule field, what people normally use, the unit people normally use are piconewtons, so that's 10 to the minus 12 newtons. And so this is 120 piconewtons. How big is that? What's the significance of that? Is that a big force and a small force? It's a huge force. It's too big, in fact. The polymerase doesn't generate that much force. That's enough to start ripping proteins apart, in fact. So let's compare it to some numbers that we know. Well, I'll compare it down. Let's compare it to some numbers that we know down here. It turns out that if you take a piece of DNA, you grab it here, and you grab it there, and you can actually do that experimentally and pull on it, then up to a certain point, the DNA just sort of extends out. It was originally in some kind of random coil. You start pulling it out until it's more or less straight. You keep going. When you get 6 to 60 piconewtons, you straighten the DNA out into what's called sDNA, which is a straight ladder with, you pull the helix right out. It becomes a straight ladder like this. And so 120 piconewtons is twice the amount of force you need to absolutely pull the DNA right out. Actually, it's probably enough to just pull the DNA strands apart, in fact. So it's a lot of force on the biochemical scale. Another comparison, here are a whole bunch of other machines that generate forces. Some of them are actual motors. Kinesin, we looked at, that's a transport motor that moves cargo along microtubules. Myosin is the motor that's in your muscles, in huge numbers in your muscles, in order to generate every force by every muscular being there is, every eukaryote. There are also versions of myosin that walk along actin filaments. Both of those generate much less force, 7 piconewtons from kinesin, between 5 and 7, even less, for myosin. RNA polymerases have been measured, and the forces vary. Again, these numbers are hard to measure, but it's 15 to 20 piconewtons, quite a bit less than the 120 I just calculated. DNA polymerases have been measured, but the number is pretty soft, 50 piconewtons, though. DNA polymerases do generate a lot of force compared to all the others, and you can kind of see why. Well, basically because they have a short step. And then ribosomes, believe it or not, moving on messenger RNAs, have also been measured, and they generate maybe around 13 piconewtons. Again, these numbers are fairly uncertain, but that's what we have right now. Why is that so? Why does the DNA polymerase generate much more force than a motor? You would think a motor would be good at generating forces, but the motor isn't optimized for generating forces. Kinesin takes a big step, an 80 angstrom step, compared to the 3.4 angstroms for the DNA polymerase, which means that the amount of work that it's doing, which is this 7 times this 80 and then converted to kilojoules per mole, is about 34 kilojoules per mole, and the delta G0 for ATP is 31 kilojoules per mole. Now, inside the cell, it's more like 50 kilojoules per mole, so this isn't quite the same as the delta G for ATP under its actual true conditions, but it's still more or less the amount of energy available from one ATP, so the kinesin is probably at least partially optimized to use the energy efficiently. Here in DNA, it's throwing away a bunch of energy, at least if what you want to do is do work. Of course, the polymerase isn't trying to do work, it's trying to replicate the genome. It's got a different function, but it's much less efficient. The work it does is only about 10 kilojoules per mole. Likewise, the RNA polymerase is even less efficient, only about 4 kilojoules per mole. All right. So you get a feeling for how big this 120 piconewtons is. It's really huge. It's a certainty that DNA doesn't generate that much force. So let's look at some other possibilities. Suppose we don't use the energy from the whole process. It has to be one step in the process, and most of the energy is going into other steps. So how about the fingers closing? Delta G there is only about minus 16, not minus 25. That gives you a maximum force of about 75 piconewtons. That's getting closer. Suppose it was on the translocation step. That's a delta G of minus 11 kilojoules per mole. And now that's getting more realistic. It's around 50 piconewtons. That might actually be true. It's very weak evidence and circumstantial, but it suggests at least that maybe the force is generated on the translocation step. The fingers closing is another good step, though. All right, with that, I think we'll stop because I think we are getting close to the end of time, so we'll pick this up next.